So this is a wonderful workshop. Thanks a lot to the organizers. Pleasure to be here. I want to frame this talk in the spirit of Simone Pigolotti's Hydra project. So where basically, I think in essence, you took a classical physics problem, sort of the absorption of finite-sized particles on a surface, and you put an element of biology in there, which is growth, and then that transformed the problem. Yeah? And I think that's sort of a little bit how I can view on this body of work. So you have a basic sort of general physics problem. That's you have some objects with well-defined properties and some boundary condition, and you have some objective function, maybe a Hamiltonian or whatever, that depends on the position of the objects, and then you try to find the optimum arrangement. And then there's many questions you can ask about the optimum arrangement. I mean, some examples of that go back to like really old problems, like the Kepler problem of stacking cannonballs in a space-saving manner. And Kepler apparently hypothesized in the 17th century that the best solution is the FSC lattice, and it took until 2005 to actually mathematically prove that. Yeah? And this kind of packing and arrangement is still interesting these days, absolutely. There's for non-spherical particles, there's some really surprising solutions that are not always ordered lattices, but non-periodic structures. So, but then biology, usually you have function. Yeah? So particles have a job to do. For example, you have catalytic abilities. Yeah? So if you have objects that perform catalytic reactions like converting A to B or B to C, then you effectively have some input substance which is converted to output in a multi-step reaction, let's say. And there are questions about how to place those catalytic particles in such a way that you, for example, optimize the output. So we have, as a human race, sort of figured out that it's good to place machines in an assembly line a couple hundred years ago, yeah, and there are sort of principles of optimizing productivity, such as division of labor, sophisticated machines, and you bring them together and you form assembly lines. And I guess we can argue that biology has discovered all these things long ago. Yeah? So we have division of labor between cells, and we have sophisticated machines, and they are brought together by co-localization mechanisms. And we don't always know what they optimize, but presumably evolution sort of tuned them, yeah, to optimize some functions. So I just want to argue that this happens on many different scales. Yeah? So in a sense, you can view an individual, let's say, bacterial cell as a catalytic object because it sort of absorbs certain things and then produces certain things. I mean, this is an example here of a filamentous organism called Anabaena, where this, in principle, single-celled organism manages to differentiate two types of cells, the nitrogen fixing cells and the carbon fixing cells, and together they can do this feat of basically taking their nitrogen source out of the air and also their carbon source out of the air, and then they basically share the metabolic products of these. Yeah? So that's on the cellular level, and you have within cells subcompartments, yeah? And these subcompartments, like the carboxysome, which is responsible for carbon fixation, that's a cellular subcompartment, and these are also arranged. Yeah? So you can view a subcompartment as a catalytic particle in a sense that gets input and generates output. And even more prevalent on the molecular level, you have these beautiful complexes that consist out of enzymes. This is one example. It's the famous pyruvate dehydrogenase complex, or PDH complex. This is in metabolism in each organism that we know of, including ourselves. This sits in the metabolism, like between the TCA cycle, the citric acid cycle, and the glycolysis pathway, sort of directly at the interface. And this object is responsible for converting pyruvate to acetyl-CoA. And it consists out of 3 different enzymes, and it's a 3-step reaction. Yeah, so that's one example. I mean, there are lots of details here which I'm not mentioning, but in the spirit of arranging catalytic particles here, it's a beautiful example. And then also on the synthetic side, yeah, so in bioengineering, and what people can do in the labs is they can fabricate things with enormous spatial resolution. So there are different techniques like using AFM tips to place single molecules on very precisely with nanometer resolution on surfaces, and then place enzymes that work together, or you can use DNA origami, or basically existing scaffolds that consist of big protein complexes, and you attach specific enzymes to them. Yeah? This is all possible. And this has even an impact. Yeah? So Jay Keating's group, for example, they use protein scaffolds to optimize metabolism within cells to produce things that cells normally don't produce. And that has a big effect. So in this case, for example, they get a 77-fold enhancement of the production. Okay, so I want to talk a little bit about the physics aspect of this, and the general theme that we see here is, I would say, that you have spatially orchestrated reaction kinetics, where you have reactions happening not everywhere homogeneously in space, but just at specific locations, and those locations are fixed. And you can, in principle, arrange them in different ways and then compare different arrangements. I'm going to, of course, so let me manage expectations. So this is mostly a theory talk. Yeah? There's practically no data in it. Yeah? So I just thought this fits to at least some of the themes in the conference, although there's no true rare event statistics, but we have sort of a bet-hedging phenomenon that I want to bring across here. So to just introduce like a spherical cow model here, let's say we treat an enzyme as a spherical finite particle, and then we have the small molecules that they convert. I treat them not as finite-sized particles, but as a field, yeah? So concentration field. The minimal system that's interesting in our context here is that you have 2 different enzymes, so 2 reactions. The substrate is converted by the first enzyme to some intermediate, and the second enzyme to a product. And let's say we have finite resource. We have a finite number of these blue enzymes, a finite number of these orange enzymes. And you get the task that you get some arena that supplies from the outside the initial substrate, and that absorbs also the final product. And you can place these enzymes in the arena and move them around. Yeah? And then for each placement of the enzymes in the arena, we figure out the steady state metabolic flux, yeah, that is generated from this arrangement. So you can see here, for example, a visualization of a particular arrangement of these blue and orange enzymes, and what is plotted in the gray color code here is the concentration of the substrate. So you basically see that the substrate is depleted by the blue enzyme. Yeah? So the orange enzyme just acts as a reflecting boundary condition for that. It doesn't interact with the primary substrate. but then the intermediate that is generated by these blue enzymes, so the intermediate concentration is highest in the vicinity of these blue enzymes in the steady state, and there's a gradient to the absorbing boundary condition on the orange enzyme that takes those intermediates in. Yeah? So this is yes, yeah. Yeah. So this is on the next slide. Yeah. So basically, we put these enzymes in. So in this case, a two-dimensional arena could be a three-dimensional arena. We'll talk about different things, but it's kind of a d-dimensional sphere around it, yeah, where there's a fixed boundary condition of substrate concentration. And in the simplest case, also this same boundary is also absorbing to the intermediate. Yeah? And that is something that's really important. I need to emphasize that. Yeah? So this intermediate product that is formed somehow needs to have a way to get lost. So either to the environment, that it gets absorbed somewhere, or there's a competing pathway. There's some other enzyme, some other thing, or like a spontaneous decay of the intermediate product. And that's typically present in all the natural cases where enzyme localization happens, is that there's a competing pathway that can happen to this product or intermediate product along the chain. Okay. Yes. Well, in general, there are different parameters. In some of the plots that I show, I just, for simplicity, assume it's the same. But no, in general, every enzyme has a effective reaction rate constant. I was just going to get to this reaction rate. So basically, the nice thing of this simple model is that you have only one dimensionless parameter for each enzyme, as long as we work in the low substrate concentration regime where saturation is not yet relevant. There, basically, we have the association rate of binding of the substrate to the catalyst. Yeah? Then there is the radius of the catalyst, the diffusion constant, and the surface area. And this expression is true regardless of the dimension. So for two-dimensional enzymes, three-dimensional, it's always dimensionless because of the 1 over volume of the association rate. And this single parameter would exist then for each of these reactions. Yeah? Yes. No, so here I'm assuming that they're just placed and the reaction only happens at those. So the enzymes are just positioned. Yeah? In some circumstances, yes, yeah. But if you have, for example, a metabolic, like a protein complex, yeah, that has a fixed stoichiometry, a fixed, geometry, there they have precise locations. Yeah? Of course, the protein complex as a whole can move. Yeah? Oftentimes, we have also association of catalytic particles to surfaces. Oftentimes, those surfaces are sources for metabolic things. So I will, yeah. So, but for the moment, for the physics problem, it's just fixed. Yeah. Well, basically, product is then assumed to be consumed further by the living organism. That is, we don't need this explicitly because we basically just measure the flux of generating product and we assume it disappears. But we don't have a physical description of product. Yeah. So since that is usually in practice not happening. Okay, so first thing, you can just play around with this. You can say, well, I just randomly place enzymes in the arena and look at the output. So basically look at the total flux, reaction flux that is generated by a specific arrangement, and then generate a large ensemble of different arrangements, just random arrangements. So then you get basically for one parameter set, so let's say you take 30 enzymes of each type, you take a particular reaction rate constant that in this case is assumed to be equal for the two enzymes, then you get a probability distribution function for the final output flux that has some mean value and some variance. And these quantities, the mean value and the variance, they depend basically on n, on alpha, and mostly on those things. Yeah? And so from studying these ensembles, random ensembles, we can extract some features like what determines the output of the system. Yeah? And I'm not going to go to the correlation analysis and so on with, for example, minimal distances between the two different enzymes, mean distances, stuff like that. You can look at all kinds of observables. You can treat this as a kind of experimental computer experiment. And I just want to quickly summarize the lessons that you observe from this correlation analysis. Main lesson is that there's two regimes. Yeah? So if the reaction rate constant, this effective dimensionless number, is small, which means that we are in the reaction limited case. Yeah? So it's the total flux is really limited by the not by the transport, by the reaction. And then in that case, you get a higher flux from arrangements that where the catalysts are closer together and they're closer to the center of the arena. Yeah? So the arena is the circular geometry where we put them. Yeah? If you're in the diffusion limited regime, yeah, so that corresponds to, because alpha has the diffusion constant in the denominator, yeah, that corresponds to large alpha. You get the highest flux from arrangements with catalysts that are further apart from each other, and they're closer to the and to the periphery of the arena. Yeah? And then there's some intermediate regime where with intermediate values of alpha, where the mean separation of the enzymes really seems to be irrelevant, but there is a big advantage of pairing always first with second enzymes. So whether it's good or not to cluster the enzymes together depends really crucially on this parameter alpha. Yeah? Yes. Wait, so self-averaging in this context would mean I mean, so basically this is sort of the degree of self-averaging then. Yeah? I mean, for a finite number of particles, I mean, each of these corresponds to different arrangements. Yeah, I just rain down, yeah, just like Simone with this adsorption problem, I just rain down enzymes here on a surface and then compute for each arrangement the flux that they produce, and then this gives a certain flux, yeah, and this has a width and a mean. This is a disorder ensemble. Yeah. Yeah, I mean, so right. I mean, so the variance, of course, decreases relative to the, variance, the standard deviation, the coefficient of variation decreases as n goes to infinity. Yeah, but we are interested in small numbers and finite systems. Yeah, Oded. Yes. Yeah. Yeah. Well, so I'm going to get to maybe to some analytics. I just wanted to illustrate the phenomenology a little bit. Yeah. And so these are the lessons here. Yeah. And so these were just random arrangements. Yeah. Of course, you could also say, well, let's put them in particular sort of arrangements that make sense. Yeah. Like, for example, making pairs or making big clusters, like you were saying, or making certain complexes with certain stoichiometries, and then you can compare them and look which is better in which regime. Yeah, so then we basically get performance of different strategies of placing enzymes with respect to each other numerically here in 2 dimensions and 3 dimensions. Yeah, and you can see that in the regime of that is reaction limited, yeah, so small alpha, basically clusters always win. Yeah, so these arrangements are really, really good, and that can be a factor of 10 in 2 dimensions. It can be a factor of 100 in 3 dimensions. Yeah, so that makes a big difference. For large alpha, clustering is actually bad. Yeah? So there is really a intersection between these different strategies. Yes, yes. What was the random part? Yes. Yes. Yeah, exactly. Just in the beginning, this random arrangement, since we only do a couple thousand, we never get even close to the optimum. Yeah. So these are much better than random. And here, this is kind of a phase diagram of the optimality of the system. Yeah. So let's say you vary the reaction rate constants of the first reaction and the second reaction, and have a fixed number of enzymes, and ask, well, for what combination, and what is the best strategy of placing the enzymes? Yeah? So then you roughly get this diagonal line that says if both reaction rate constants are not too large, yeah, or if this along the diagonal here, then clustering is best. Yeah? And then the relative speed of the two enzymes determines the stoichiometry of the cluster that you want to have. So, for example, a 4-to-1, a 3-to-1, a 2-to-1, and so on. Yeah? So this makes intuitive sense, I would say. Right? If you have an enzyme A that is 4 times faster than enzyme B, then you put 4 of them together. And then sorry, so that was the solution for large alphas. Yeah, so that's these complexes. Yeah, and cluster is this strategy that's always the best. Like lump everything together closely in the reaction limited regime. Okay, so the minimal thing that I would like to get across in this talk is the basic physics that's behind this. Yeah, and part of that can be understood in a very simple toy model. So this is really old work, but I just want to bring it up to explain some of the physics that's in here, and that also relates to this idea of bet-hedging in molecular systems that promised. So again, we do the simple two-step reaction, and we have an intermediate product that is described by a concentration field. But now I also describe the enzymes by a concentration field. Yeah? So I'm smoothing. It's like and we're doing, for simplicity, for now, just everything in 1 dimension. Yeah? This can be generalized later, but it turns out it doesn't change things much. Yeah? In contrast to many diffusion problems where it's a very important whether you're in 1, 2, or 3 dimensions, because the return statistics depend sensitively on that. Yeah? But here, since we always have a loss pathway, yeah, for example, you have on the right-hand side here a absorbing boundary condition. On the left-hand side, you have a source of the intermediate. And then the question is how to place enzymes in the system, a finite amount of enzymes in the system, such as to optimize the total product flux. Yeah? So mathematically, this can be written down as a reaction diffusion equation. In the simplest case, it would just be linear. Yeah. But it's sort of not the standard type of problem that you solve because now you have this function e of x in here in the reaction term, which just has a condition that the total amount of enzyme is fixed. Yeah, so the integral over e of x is finite. Yeah, in a fixed number. But now you can choose this function freely in order to optimize the flux. Yeah? And the quantity of interest is this total reaction flux in the steady state, which is just a simple integral of the distribution of the enzymes over the solution, the steady-state solution for the intermediate product. So you have to find, for a given E of x, you have to find the steady-state solution of this PDE here. So that gives you some intermediate distribution, and the output flux is this integral. You're trying to optimize that. Yes, yes. E is the second enzyme. Yeah. So here we are assuming. Yeah, the first step is sort of sitting at the boundary here. So I'm simplifying the problem to the absolute minimum here. Yeah, just to get out the physics. So it's here. Yeah. And the first thing you can do is to just, let's put all the enzymes that you have at the source of the intermediate, which is at the left edge. Yeah? So I choose for E a delta function, yeah? And then I get a output flux relative to the input flux. That's the red curve here. And the other extreme limit would be we take all the enzymes and smear them out evenly over the system, and then we get the blue curve. Yeah? And what's interesting is that these two intersect, so that means even in this toy model we have the same phenomenon, namely that clustering is good and superior for slow reactions, and for fast reactions the uniform spreading out is superior. And what's the physics of this? So you can understand the physics of that by treating the transport and the reaction as sort of two different aspects of the problem. Yeah? So let's say we generate all possible diffusive trajectories, and this can be generalized to advection and diffusion, that start from the left edge and they end on the right edge. Yeah? So you have a reflecting boundary with influx here, and you have a absorbing boundary over here. So you generate all possible trajectories, and along each trajectory of the intermediate particle, particle can be killed, yeah? Killed by a reaction, basically. Yeah? So the entire probability of a reaction happening, or the flux then also, would be the sum over all possible trajectories, the probability for a trajectory times the probability of a reaction along a trajectory. This probability of the reaction along a trajectory, that depends on the enzyme arrangement, whereas the trajectories themselves, they're completely independent of the enzyme arrangements. Okay, and that brings me to a term or a concept, let's say, that we named enzyme exposure. Yeah? So you can say, well, if you have a particular trajectory of a intermediate particle, then during its path through the system, it sees a time-dependent enzyme concentration, and that can be summed up by an integral over time until the escape time of the trajectory, which is like a total enzyme exposure that it sees over its lifetime, yeah? Over its time to escape. And different enzyme arrangements will produce different stochastic distributions of enzyme exposure. So basically, we get that the total reaction flux is a convolution of these two functions. So we have the reaction probability for a given enzyme exposure that is just given by this simple function, namely 1 is not reacted minus, so the probability of having reacted is the probability of not having reacted minus exponential of reaction rate times enzyme exposure. So this is very similar to the, yeah. But then we have this probability distribution for what is the enzyme exposure, what is a certain enzyme exposure, how often does it occur? And that depends on the arrangement of the enzymes, but it doesn't depend on the reaction rate. Okay? And now we can try to understand the the transition that happens in this term. Yeah? So, for example, so the key information is that having a cluster at the source gives you a higher probability of having high enzyme exposure because the diffusing particle spends most of its time close to the source and less time away from the source. So basically, we have a distribution of enzyme exposure for a cluster that has finite probability to have essentially no enzyme exposure. So those are these extreme trajectories which go like directly to the right, directly escape from the source. We talked about that yesterday in a different talk. But then it has this long tail of high enzyme exposure. Whereas a uniform distribution, it's impossible to not see any enzymes because enzymes are everywhere. But the probability to get a lot of enzyme exposure is lower. So these two distributions for the two different arrangements, they are fixed. But what matters is how the enzyme distribution, enzyme exposure is weighted by the reaction probability. And that depends on the reaction rate. So if you have a low reaction rate, it's particularly important to have high enzyme exposure. And for high enzyme exposure, you have this long tail of the clusters, which get weighted, it gets linearly weighted much more. So it's beneficial to cluster. But for high alpha, so fast reaction, basically, it doesn't really matter how much enzyme exposure you have. You just want to avoid having very low enzyme exposure. So it's really like you've reached maximal probability of reacting at very low levels of E already. But then there's this initial regime. And there it only matters that the uniform distribution has this advantage that it never gets no enzymes, never gets to see no enzymes. So that's basically, it's a bet-hedging phenomenon. And that's my only link basically to the theme of this workshop. But I want to, so basically the insights from that toy model is that co-localization of enzymes increases the efficiency for slow reactions. Distributing enzymes is good for fast reactions. The optimal arrangements is a mixture of both strategies, which is molecular bet-hedging. And this enzyme exposure is useful to understand this. Okay, so let's now go back to the full model. So we have this phase diagram of the optimal arrangements. We see the basic phenomenon along this axis. So we see the transition from clustered to non-clustered configurations. But then let's also look at like the true optimum. If you have a certain, there are additional effects in this discrete particle system that we don't have in this mean field description. I probably need to speed up. So and there's two things that this mean field description ignores. One is a shielding effect, namely that if there is, for example, an arrangement where you have an enzyme that is inside a group of other enzymes, then it gets less access to substrate that's coming from the outside. And there's also a confinement effect that things that can be trapped inside. And the interplay between these two things leads to like truly global optimal configurations. And yeah, I get the hint. So I skipped some things, and I want to show you sort of optimal 3D complexes of enzymes for this minimal model. This is the way they look like. This is the case where we have one of the first enzyme and a varying number of second enzymes, so 6, 7, 8, 9, and so on, 12. And now, those of you who know electrostatics and have heard of the Thomson problem, might be interested in hearing that the configurations that optimize this enzymatic problem happen to be identical to the optimal solutions of the famous Thomson problem, which is the problem where you have equal charges distributed on a sphere and the question is, how do they distribute themselves in order to minimize the total electrostatic energy? And for a reason that we cannot prove yet, empirically, the solutions are identical to these two different problems. Yeah, so the Laplacian is definitely shared by both, yeah. But then we have different boundary conditions here, and that is something that confuses me. And we don't have a potential here and so on, right? So, but this happens to be, yeah. I mean, the reason must be the Laplacian, right? Then you may argue that real enzymes have catalytic sites that affects the outcome of the. Anyway, I'm just going to stop. I mean, yeah. Yeah, there's lots of things that you can do. Yeah. Okay. Yeah. So to conclude, yeah. So it's sort of an element of biology that enriches physics, I would say. Biology can use dynamic clustering as a regulation strategy, something I didn't talk about. Then you have real carboxysomes that have different properties in nature. You can understand some of that using this model. And the problem can also be treated analytically using variational, that might not surprise you, calculus using, and the nice thing is that this links to economic principles. But last but not least, let me thank the people who did this work. This was real fun to do it. They work with extremely talented postdoc at the time, Philippe Tostevin, and then PhD students here, Alex, Florian, and Giovanni, and then external collaboration. Thanks a lot. Sorry for being over time.